Hi, good day to all participants. It's a great pleasure to meet all bioprofessionals attending BioKorea 2021. And I would like to thank to organizers for organizing this great convention and inviting DEM Pharma to be a part of it. My name is Necla Sena Korkmaz. I am molecular biologist and I took my master's and subsequently postmaster's degree in biotechnology. I worked at Janssen Biologics in the Netherlands, and my role was characterizing the glycan profile of two important commercially antibodies. And I have been working in DEM Pharma since 2014. As you mentioned, I'm a biotech R&D manager. And I would like to, I would like to give a brief information about my company. DEM Pharmaceuticals is a young, dynamic, and fast-growing pharmaceutical company with its origin as a prosperous pharmacy. Our mission is to use our resources effectively and to continuously increase our competitive quality and to follow technology and open to further developments. And our vision is to become one of the leading companies in Turkish pharmaceutical sector and prioritizing the importance given to patients' health and to the doctors and pharmacists. As I said, we are a growing company and we grow day by day. As of today, DEM Pharma has almost 300 employees all over Turkey. We have 4 product lines, hospital and specific products line with 13 brands such as Hibor, SLN, and etc. We have hematology and oncology products line with 12 brands such as Leucostim, Thromboredictin, etc. We have blood derivatives product line with 9 brands such as Gamunex C. And also we have nephrology products line with 6 brands such as Eparon, Calderol, and etc. DEM Pharmaceuticals holds marketing rights of high potential pharmaceutical products from worldwide companies, which are experts in their respective domains. And these companies include Donga, Rovi, Grifols, and etc. DEM Pharma has a large but specific product portfolio, and it has a rich but supplementary pipeline. In this picture, you can see a small but highly motivated and highly efficient R&D team. We recently hired 2 more colleagues in our group, and now we have 10 scientists, 10 successful scientists in our R&D team. And I would like to introduce my team. So this beautiful lady in the picture is our scientific division director, Esra Üstünyaz. And this lady is our analytical lab manager, Nurgül Akçın Önel. And also here you can see our senior scientists. We have the only guy here, Osman Baş, and also Sıla Tümen Erdem. In our group, we have 2 scientists with a PhD degree, and as I said, I have a professional doctorate in engineering title. 50% of our team has master's degree, and the rest has bachelor's degree with a long time experience in pharma industry. In DEM Pharma, we have 3 full-fledged laboratories, and these include cell culture laboratory, analytical laboratory, and microbiology laboratory. Our analytical laboratory has an accreditation from Turkish Standards Institute. In our laboratories, we have several basic and high technology lab equipments, and we have, for example, Ember 15 microbioreactor system. We have BioCore T200, PA800 capillary electrophoresis system, Q Executive Plus Orbitrap LCMS system, and HPLC systems with 4 different detectors. There are some pictures from our laboratories. In the top row, you can see some spots from our analytical laboratory. In the middle row, you can see some pictures from our cell culture laboratory, and in the bottom row, you can see some pictures from our microbiology laboratory. Our R&D team focuses on three main R&D activities, and these include biosimilar development, nanosimilar development, and also peptide derivatives formulation and analytical development. So first, I would like to talk about our biosimilar development studies. This is our step-by-step approach to develop biosimilars. So we start our studies by the determination of reference molecule amino acid sequence. And this amino acid sequence is codon optimized for higher expression in mammalian cells. And plasmid vectors appropriate for the study are designed by our R&D team in-house. We outsource synthesis of synthetic cDNA. When the vectors and also the cDNAs are ready, we insert the cDNA into the designed vector in-house, again by our R&D team. And the vectors are produced in E. coli cells and subsequently isolated and purified in our microbiology laboratory. To ensure proper integration of the vectors into the host genome, we always linearize our vectors. And meanwhile, we prepare our host cells for the transfection in our cell culture laboratory. When everything is ready, our cells, our vectors are ready for the transfection, we start transfection studies. And we perform several transfections to find the desired stably transfected cell line. And when we say yes, we found it, the stably transfected cell, we continue with this pool with the clone selection. So we perform several rounds of clone selection, and when we are happy with our highest producing clones, we continue with the next step, which is process optimization. And we optimize our process in the microscale in Ember 15 microbioreactor system. And we say, yes, okay, we are happy with the process. We transfer our process and our cells to the next stage, to 1 liter bioreactor production. And this is followed by 5 liter production and 50 liter production. And also we perform a comparative protein characterization in all the steps, starting from the top clone selection. So now we have 2 ongoing biosimilar protein development projects. One of them is coded as DEM Bio 1, and the other one is coded as DEM Bio 2. DEM Bio 2 is funded by the Scientific and Technological Research Council of Turkey. DEM Bio 1 is a biosimilar of a molecular antibody which is indicated for colorectal cancer, and DEM Bio 2 is indicated for non-Hodgkin lymphoma. By following the stepwise approach that I showed in my previous slide, we successfully came to the production in lab-scale bioreactor stage for both projects. And also, we showed the biosimilarity of candidate biosimilar maps produced in 1 liter bioreactors to the reference molecule, which I will be talking about soon. So, some biosimilar comparative assays that are performed in our labs by our R&D team. For example, we perform STS page and Western blot to qualify our maps. And also, we perform intact protein mass and subunit mass detection by LCMS system. And we detect the isoelectric point of our products and also to the reference protein by capillary electrophoresis system. Our IgG's purity is determined by a validated size exclusion chromatography HPLC method and also capillary electrophoresis. And also we perform LCMS analysis to characterize the glycan profile of our products. And last but not limited to, we perform post-translational modification detection such as lysine truncation or pyroglutamate formation, again by LCMS system. So now I would like to show you some biosimilarity assessment results. In this picture, you can see the deconvoluted spectrum of reference proteins and the reference protein, and also our biosimilar proteins produced by two different clones. And as you can see, they are all, they gave a peak at 147 kilodaltons. So they are massively very comparative. And also the post-translational modifications, they are very comparative. And this is the subunit mass analysis picture. Again, we can see the deconvoluted spectrum. Here in this picture, we see the mass of our light chains. And again, you can see that they gave a peak at the same point, and also pyroglutamate formation can be seen for all the samples that we performed. Also, as I said, we can determine the isoelectric point of the samples. In this picture, in this graph, you can see that the PI of our proteins are very similar to reference protein. And if we go into the details, these are the printout from the PA 800 plus system. Here you can see that we get the PI of the reference protein, and it is 9.2, and also our protein's PI is 9.2, and also the other one, the other biosimilar candidate, it's 9.2. So our second focus is nanosimilar drug development. Nowadays we have three nanosimilar projects. One of them is coded as DEM Nano1. It's a nanosimilar of an anti-infection drug. And the DEM Nano2 and DEM Nano3 are the nanosimilars of two different anti-cancer drugs. So DEM Nano1, it's a liposomal product, and we produce this nanosimilar by using membrane extrusion and lyophilization technology. And in this picture, you can see in the middle the reference product, and the rest are the products that we produce. They are from different batches, and visually, as you can see, they are very similar. And of course, we performed several assays to show the nanosimilarity, and for pilot scale, physicochemical characterizations, in vitro biological activity studies, and in vivo animal studies of DEM Nano1 are done. DEM Nano2 and DEM Nano3 projects are still in progress. So lastly, I would like to talk about our manufacturing facility, production facility in Yalova. The building of the facility has been under construction since 2018. And the total land area is 30 decare, and the total closed area is 17,500 square meters. And we are aiming to take our GMP approval till the end of this year. So there are some pictures from our facility. So thank you for your attention. And if you need further information, please do not hesitate to send an email to this address. Thank you.